Imagine a world where new drugs are developed in months, not years. A world where pharmaceutical breakthroughs are faster, safer, and far more accurate. Now picture this happening without a single mouse, monkey, or dog suffering in a lab. Sounds like science fiction? As of 2024, it's rapidly becoming reality. But what if I told you that the very foundation of modern drug discovery has been crumbling for decades, and that billions of dollars are wasted each year because of a stunning 95% failure rate in human clinical trials after promising animal tests? What's behind this catastrophic waste, and how can cutting-edge technology finally break the cycle? Stay with us, because today we're dissecting the revolution quietly unfolding in biotech labs across the globe. The story involves robotics, artificial intelligence, and living human tissues grown on demand. The stakes? Nothing less than the future of medicine and the end of animal testing as we know it. Why do so many drugs that work in animals fail catastrophically in humans? The answer is equal parts ethical crisis and technological challenge. For centuries, animal testing has been the default path to medical breakthroughs. From penicillin to statins to COVID-19 antivirals, virtually every pill you've swallowed began its journey inside a rat, rabbit, or primate. But here's the brutal truth. Animals are not humans. Their immune systems, metabolisms, and gene expressions differ in ways that can make effective drugs look like failures, or worse, deadly poisons, when finally tested in people. Famed researcher Dr. Thomas Hartung sums it up. AI is a tremendous revolution in dealing with information, redefining the speed and scale of scientific discoveries. But for decades, information from animal models has been, frankly, misleading. Take chronic diseases, the kind that kill millions each year, from cancer and diabetes to Alzheimer's. Mice don't get these diseases the way we do, so we've been flying blind, spending years and billions on drugs that look promising in a cage but fall apart in a hospital. Pharmaceutical companies know the numbers. 95% of candidate drugs fail in human trials after passing animal tests. That's not just bad luck, it's a systemic failure, costing the industry, and ultimately, patients, untold billions and lost decades. The pressure is so severe that some startups now refuse to touch drug development unless they can work directly with human data. This is where the extraordinary story of Vivodyne enters. Backed by $40 million in fresh funding from Khosla Ventures and others, this U.S. biotech is taking a radical new approach: eliminate animal testing entirely. Their secret? Replacing animal models with thousands of living, fully functional human tissues, all grown and analyzed by robotics and AI at a scale previously unimaginable. Let's break down what that really means. Vivodyne's new 23,000 square foot facility in South San Francisco will soon be producing over 10,000 individual human tissue experiments in a single robotic run, far eclipsing traditional lab capabilities. Forget mice in cages. Imagine robotic arms nurturing miniature livers, lungs, bone marrow, and even placenta tissues, each hooked up to sensors and AI systems that read every reaction in real time. Dr. Andrei Georgescu, Vivodyne's co-founder and CEO, puts it bluntly: we cannot effectively discover how to make humans live well beyond 100 years by testing in mice with lifespans of 1 year. Now Vivodyne is enabling large-scale research on lab-grown human organ tissues to produce massive amounts of new data and, hopefully, actionable insights from the pharmaceutical industry. The scale is breathtaking. Using robotics and AI, Vivodyne can grow and test over 100,000 different whole human tissues automatically within 2 weeks. That's an experimental throughput that no animal-based lab, no matter how vast, could ever match. Vinod Khosla, the legendary tech investor who led Vivodyne's latest funding round, says it simply: robotics and AI are already starting to fundamentally change the healthcare landscape, and Vivodyne is pioneering the way there. This revolution isn't happening in isolation. Across the biotech landscape, new companies are racing to capitalize on a historic policy shift. The U.S. FDA Modernization Act 2.0, passed in recent years, now allows drug developers to use non-animal models for safety and efficacy data. That's a tectonic change. For the first time, there's a regulatory path for drugs to reach the clinic using only human tissue and AI data. It's not just Vivodyne making waves. Pioneers like Emulate, Mimetis, and Inspiro have pushed the boundaries with organ-on-a-chip and organoid technologies. In 2024, for the very first time, Hesperos helped pharma giant Sanofi receive FDA approval to move a drug to clinical trials based solely on data from organ-on-a-chip models. Meanwhile, startups like Frontier Bio have successfully 3D bioprinted lung microtissue, while Chorus AI and others are using machine learning to predict toxicities before a single patient is dosed. Emily Trunnell, a neuroscientist at PETA who spent her career fighting against animal testing, is surprisingly optimistic. Non-animal methods consistently predict human response better than animal models, offering areas where they can replace animal testing. After centuries of ethical debate, the technology finally seems ready to deliver on that promise. But just how accurate are these new methods? Can a vat of lab-grown human tissues steered by machine learning really replicate the infinitely complex reality of a living person? This is the crux of the controversy and the cutting edge of biotech. Let's zoom in on Vivodyne's innovation. Their system can culture more than 20 different types of human tissue, encompassing liver, lung, bone marrow, lymph node, placenta, and beyond. Each tissue isn't just a blob of cells, it's engineered to function like the real thing, supporting high-resolution multi-omic data collection. That means imaging, single-cell RNA sequencing, proteomics, all the cutting-edge tools scientists use to decode how diseases unfold and how drugs intervene. With these capabilities, pharmaceutical companies can finally tackle diseases at the tissue level, modeling conditions as diverse as cancer, fibrosis, autoimmune disorders, and infections. Instead of inferring how a human liver will react to a new drug by watching what happens in a mouse, researchers can now observe direct human responses. The fantasy of personalized medicine suddenly feels within reach. The scale of data is mind-boggling. A single robotic run can analyze over 10,000 tissues, creating a trove of information orders of magnitude larger than traditional preclinical labs. AI algorithms sift through this ocean of data, picking up subtle markers that might otherwise be lost. As Dr. Hartung notes, the combination of AI and human tissue platforms represents a tremendous revolution in scientific discovery. Yet despite this flurry of optimism and funding, the transformation is far from guaranteed. Precision is everything in drug development. While organoid and organ-on-a-chip systems are exciting, they are still just models, simplified representations of a vastly more complicated reality. There are deep uncertainties about whether these platforms can capture the full picture, especially for complex, chronic diseases that play out over decades. On the regulatory front, old habits die hard. The FDA may encourage alternatives, but implementation is patchy. Many pharma executives remain skeptical. Can these new models survive the scrutiny of global regulators? Will insurance companies and hospital groups trust the data? There's also the challenge of validation, proving beyond doubt that a robotic, AI-driven tissue experiment actually predicts what happens in real patients. Jan Lichtenberg, CEO of Inspiro, puts it in context. 3D Insight, human liver microtissues represent a promising step toward reducing attrition and improving safety testing. It's a step, but not yet the finish line. Let's not forget, the economic incentives are massive. If these new platforms can even halve the current clinical trial failure rate, the savings would run in the tens of billions per year, and new drugs could reach patients at a fraction of today's cost and time. The race is on, and every major player wants a piece of the pie. Yet the road ahead is anything but smooth. Aside from technical and regulatory hurdles, there's the stubborn challenge of culture and inertia. Pharma is an industry known for risk aversion, and shifting away from the centuries-old animal testing paradigm requires both trust and proof. Will investors stay patient if results lag? Will government agencies harmonize their standards globally? And what about the philosophical question? Can machines, algorithms, and engineered tissues ever stand in for the complexity and unpredictability of real-life human biology? As we peer into the future, we're left with a tantalizing question. Will the convergence of robotics, artificial intelligence, and living human tissues finally slay the 95% failure rate dragon, or will new complexities emerge, threatening to derail the most promising revolution in drug development in a generation? The answer isn't just academic. It could shape the fate of medicine, industry, and millions of patients worldwide. In the next part, we'll dive deeper into the battles raging behind the scenes, from the technical arms race between competing startups to the regulatory, ethical, and commercial landmines that could make or break this seismic shift. But before we look forward, ask yourself, are we truly ready to abandon centuries of animal-based science, or are there risks lurking in the promise of the AI-driven robotic lab? Let's find out. The quest to revolutionize drug development is at a crossroads, a moment both thrilling and tense. If robotics, artificial intelligence, and lab-grown human tissues can succeed where animal testing failed, We stand on the brink of a scientific leap that could change not just medicine, but human health as we know it. But what does this future look like, and what could stand in the way? Let's imagine a world, maybe just a decade from now, where the majority of new drugs never see the inside of an animal testing facility. Instead, robotic arms tirelessly assemble human tissues, livers, lungs, even entire organ systems, inside gleaming labs. AI algorithms pore over millions of data points from these living, breathing mini-organs, predicting how a new drug will behave in a real human body with a level of nuance that even today's most advanced animal tests can't approach. It sounds like science fiction, but Vivodyne's 23,000 square foot robotic facility under construction in South San Francisco is already turning this vision into reality. This isn't an isolated case. Across the globe, startups like Emulate, Mimetis, Inspiro, and Frontier Bio are racing to create ever more sophisticated organ-on-a-chip and organoid technologies. In 2024, Hesperos even helped Sanofi get FDA approval for a clinical trial based solely on data from organ-on-a-chip models, a world first. As Dr. Thomas Hartung of Johns Hopkins puts it, AI is a tremendous revolution in dealing with information, redefining the speed and scale of scientific discoveries. But the speed of progress brings new questions. What are the risks, the opportunities, and the unforeseen consequences of moving away from animal models? On the upside, the benefits are massive. With more accurate preclinical testing, pharma companies can slash the astronomical costs of failed clinical trials, where 19 out of every 20 drugs that work in mice ultimately fail in humans. These savings could be redirected into researching rare diseases, neglected conditions, or personalized therapies tuned to your own genetic code. In fact, the very premise of longevity research, that we could one day routinely live well beyond 100, may hinge on these technologies, as Vivodyne's CEO, Dr. Andrei Georgescu, notes. We cannot effectively discover how to make humans live well beyond 100 years by testing in mice with lifespans of 1 year. Patients could also see safer, more effective drugs reach the market faster. Because these lab-grown tissues aren't just simplified models, they're constructed with human DNA, grown under automated and strictly controlled conditions, and can reproduce the complexity of real organs. From cancer to autoimmune disorders and fibrotic diseases, every unique twist and turn of human biology can finally be mirrored in the lab, without waiting years for animal data to, likely, disappoint. There's another less discussed effect, ending or greatly reducing the ethical controversy around animal testing. Each year, millions of animals are used in drug trials. Many never survive the process. If the industry truly shifts to tissue-based and AI-driven models, the era of sacrificing animals for human health might finally draw to a close. Emily Trunnell from PETA argues, non-animal methods consistently predict human response better than animal models, offering areas where they can replace animal testing. If the data hold up, we all may soon see a pharmaceutical landscape as ethical as it is advanced. However, every revolution has its challenges. The most immediate is regulatory. Drug approval processes are built on decades of precedent, most of it tied to animal data. While the FDA Modernization Act 2.0 is an enormous step forward, making it legal for companies to get early safety data from non-animal methods, there's still a lot of inertia. According to Olivier Barberin at Elsevier, you can leverage past precedent insights to potentially reduce the need for an animal study or reduce the length of the study. But leveraging precedent isn't the same as rewriting the rules entirely. Regulators worldwide must harmonize standards for non-animal methods, or else global drug approval will become patchwork, and progress will stall. The next challenge is validation. It's not enough for a robotic system to grow human tissues. Those tissues must be proven to accurately mimic the dazzling complexity of a living human. Can a bioprinted liver, nurtured under fluorescent lights, truly recapitulate the immune responses, metabolic quirks, and disease pathways of the real thing? Trials like Sanofi's show promise, but a single successful precedent doesn't guarantee universal reliability. Deep, ongoing collaboration between biotech firms and regulators is crucial, because a false positive or negative at this stage could have dire consequences for real patients down the line. And then there's scale. Vivodyne's robots may already grow over 10,000 tissue samples per run, but even that is a drop in the bucket compared to the global demand for reliable, high-throughput testing. Building facilities, training personnel, and refining AI algorithms will take relentless investment, not just from venture capital, but governments and the public sector. The prize, though, is huge: the near elimination of billion-dollar late-stage clinical trial failures and a renaissance in drug discovery for hard-to-treat and rare diseases. A final, more subtle threat: technology itself. As AI's role in biotech grows, so does dependence on algorithms whose inner workings can be opaque even to their creators. Biases in training data, statistical quirks, or even subtle programming errors could lead to missed safety signals or exaggerated benefits. Transparent, auditable AI systems must become the norm, not the exception. So what does all of this mean for you, our viewer? Why should it matter if robots are growing tiny human livers and AI is reading their cells like open books? On one level, this story is about the future of medicine. If you or a loved one have ever waited anxiously for an experimental drug to be approved, you know how agonizingly slow and uncertain the process can be. With new tissue and AI methods, that process could speed up. Safer, better drugs, brought to market faster, at potentially lower prices. The risks of unexpected side effects could drop as new treatments are tested on human tissues that actually reflect the diversity of real patients. That's a revolution with direct personal consequences for all of us. But the impact goes beyond healthcare. The very nature of scientific discovery is changing. As Dr. Hartung observed, AI is redefining the speed and scale of scientific discoveries. If AI can help us find new cures, repurpose old drugs, or even design personalized treatments de novo, the possibilities are almost limitless. Startups and researchers working on rare, orphan diseases may finally have the tools to make real breakthroughs, as the cost and time barriers are lowered dramatically. Even academic labs, long shut out by the enormous cost of animal facilities, can join the fray. There's also a growing movement for transparency and ethics in science. As non-animal models gain credibility, consumer advocacy groups will increasingly scrutinize which companies are truly embracing humane testing. This might one day influence not just policy, but consumer behavior. Imagine choosing over-the-counter medicines because they were developed without animal suffering. For students, young scientists, or anyone considering a career in biotech, this revolution opens entirely new types of jobs: robotics technicians, AI biology modelers, tissue engineers, roles that didn't exist a decade ago. If you have coding skills, a background in life sciences, or just a passion for ethical innovation, the next decade could be your time to shine. What's more, understanding these shifts lets you be an informed voice in public debate. Already, government funding and regulations are catching up to the new reality, but there will be fierce battles over standards, safety, and accessibility. Lifting the curtain on how your medicines are tested and who benefits empowers you to participate in one of the most consequential social decisions of our era. And if you're an investor or entrepreneur, the window is wide open, though not for long. The companies that adapt earliest, build strong validation pipelines, and collaborate closely with regulators will define the next era of medicine. So, let's return to our key question. Can robotics, AI, and lab-grown human tissues truly replace animal models at scale, and what could disrupt this revolution? The answer, based on everything we've seen, is yes, the replacement is well underway, but it's not a done deal. The science is advancing faster than ever. Regulatory change is catching up. Major pharma players are already redirecting resources and running key experiments on these platforms. In the next decade, the use of animal models in drug development could shrink dramatically, especially in early safety and efficacy testing. New technologies like Vivodynes are proving they can generate truly predictive, human-relevant data at a scale unimaginable just a few years ago. The challenge now is to ensure reliability, secure regulatory buy-in, and build the massive infrastructure needed for a global transition. But beware, the revolution could be slowed by regulatory inertia, technical blind spots, or even public backlash if high-profile failures occur. Transparency, cross-industry cooperation, and relentless validation are the watchwords of this new era. As we close, here's a secret fact. The very first FDA-approved clinical trial to rely solely on organ-on-a-chip technology happened as recently as 2024, using a liver-on-a-chip model. This milestone went almost unnoticed by the public, but in future history books, it may be remembered as the true beginning of the end for animal-based drug development. If you're excited, or even just curious about this new frontier, now is the time to pay attention. Subscribe to our channel for more insights into biotech's biggest breakthroughs. Comment below. Would you trust a drug tested only on lab-grown human tissues and AI? What questions do you have about this brave new world of medicine? Thanks for watching. The future of drug development isn't just about